It's my pleasure to welcome everybody to this week's edition of the EV Journal Club. And we have a guest today, Tom Whittaker, who is joining us from London in the UK. And so Tom had done his PhD in the lab of Molly Stevens at Imperial. And so what he's going to be talking with us today about is basically stemming from that PhD research that he did. And now he has moved to another location in London, University College London. And he's going to be speaking with us today about his work with MSC-EVs. So mesenchymal stem or stromal cells, of course, we all know that they have, there's been a lot of interest in them over the last few decades and hundreds of clinical trials. And what exactly is responsible for the effects of MSCs has been a matter of much investigation. So we're going to hear from Tom today about his findings on how EVs can be responsible for those effects, or perhaps other factors that may come along with the EVs. So Tom, thank you so much for joining us today, and I will hand the screen sharing over to you.
Hi, thank you very much for having me. As Ken said, I'm Tom. This work is from my PhD, where I was looking at the potential for regenerative medicine of exosomes from mesenchymal stem cells. And the focus of this talk today is going to be about some technical and conceptual challenges that I identified during this research that I think may have applicability not just to MSC-EV research, but also to any EV research where we're looking at some kind of function from the EVs. And I'm just going to start off, if I can use it, lets me. There we go. I haven't put a slide in saying what EVs are because I think we all know, but I will just briefly recap the background with MSCs. So mesenchymal stem cells are a pluripotent adult stem cell that you can get from more or less any vascular tissue. They're probably derived from pericytes, which are cells that surround and support the vasculature. In vitro, they can differentiate into several different lineages, and as Ken said, there's been a lot of excitement in using them to treat injuries where there's tissue death, because the idea being you can take stem cells from a particular patient and put them back in, and because they're the same genotype, you don't have the issues of immune rejection. And so there's been a lot of excitement for them, mostly in the context of ischemic injuries like heart attack, stroke, and kidney damage. But what was realized quite early on was that even though there was some kind of benefit from these experiments where the stem cells were injected at or near the site of injury, it couldn't actually be due to the stem cells forming new tissue, because the stem cells didn't engraft in significant numbers, they didn't differentiate in significant numbers, and the benefits, such as they were observed to take place quite quickly within the first few days. And so it was realized back in, I believe, 2005 by Whittaker et al. That it was actually due to paracrine signaling from the MSCs. So most of the reported beneficial effects were due to signaling to native tissue at or near the site of injury. And so since then, there's been a lot of work to look at the paracrine signaling mechanism that's underlying this and to try and work out what it is, and to try and work out whether it can be used as a therapeutic. And since then, from my reading of the field, there have been two almost parallel lines of research that are only really starting to come back together now. And on the one hand, ever since the 2005 paper, there's been a lot of focus on looking at conventional soluble signaling factors. So in the case of cardiac ischemia, stuff like VEGF, FGF, IGF have been implicated. For stroke, it's neural growth factors. For kidney, it's again quite conventional growth factors, things that MSCs are known to upregulate, especially in response to hypoxia. These factors have mostly been identified independently by multiple studies from multiple different labs, and I believe they have been tested independently as potential therapeutics. And then in parallel to that, there's the extracellular vesicles that we're all familiar with. And so I believe the first study was back in 2008 and was then followed up by another one in 2010. And this was the first study to really implicate EVs in this process. And so the first key bit of evidence was the observation that if you take conditioned medium from mesenchymal stem cells and use it in I believe initially a pig model and then a mouse model of myocardial infarction, you can see that you get a reduction of the infarct size with the conditioned medium. But if you pass that conditioned medium through a membrane with a pore size of 1,000 to 100 kilodaltons, you find that that activity is completely removed. So here you see that after the media has gone through these membranes, the infarct size is the same as if you treated it with saline. But what they did find is that the reduction in infarct size was still observed if you looked at the stuff that didn't go through the membrane. So from this, they concluded that there was a large complex involved in signaling, and that this large complex was the key factor. They then went on and purified it with chromatography. They found a fraction that was still active, and that fraction contained EVs, and it kind of all went from there. The reason I started this strand of research that I'm going to talk to you about is that I thought that this was inconsistent with the other literature on soluble factors. So if you think about it, what this essentially says, this initial piece of research implicating EVs, is that nothing under 1,000 kilodaltons matters. In other words, it pretty much completely rules out any contribution from soluble factors in any meaningful sense. And I thought that that didn't quite make sense to me. So that's kind of what precipitated this. And just to give a little bit of further background about why I was thinking about doing this research. Since 2008, 2010, when this all really took off, MSC-EVs have been very widely used. Lots of different injury models, lots of different sources of MSCs, lots of in vitro and in vivo work, and it kind of seems like pretty much any injury you put them to, something happens. However, my reading of the field, and I understand that this may be partly a matter of opinion, my reading of the field is that there's no consensus on mechanism. So pretty much every lab will find something different inside the MSC-EVs that are doing something. Some labs say it's this microRNA, some labs say it's that microRNA, some say it's protein complementation. And I've noticed as well that has started to be some mentioning in the literature of concerns about reproducibility. And something else that I've noticed is that although this first set of studies did an experiment where they took the conditioned medium and removed the EVs, subsequently to that, that's not really done. Instead, what people do is they take their initial conditioned medium, they go through an isolation process, and then they have an EV sample and they do something with that. And the key thing about doing that is if you don't do this initial depletion experiment, you're not actually checking that EVs are necessary. You're just seeing whether they are sufficient, whether they can do something, not whether they're required for something. And that's a distinction that I think is important that's going to come up again later. So to get on to my actual work, the first thing I did was essentially the same experiment I just showed you from those papers, where I took conditioned medium from MSCs and I passed it through a filter. But what I did differently was that I took sequential fractions of about 20 ml each, one after the other, of the flow through. Initially, I checked to see whether there were any EVs in the flow through. So you can see that there's detectable particles in the conditioned medium. There's detectable CD63. Sorry, this should be labeled. It's CD63. And there's nothing in the negative control or any of the flow throughs. So the ultrafiltration membrane is excluding EVs. And there are particles present and their diameter is about what you'd expect. However, when you go and look at the biological activity of these fractions in an in vitro angiogenesis assay, you see something that's a little bit concerning. If you look at the first flow-through fraction, in other words, the first 20 ml, it looks like removing EVs removes the bioactivity. So if you look here, this is the conditioned medium. Can you see my mouse, Kenneth?
I do not see it. Oh, I do see it. I do see it. I do see it.
Okay, cool. I wasn't sure if I was circling anything. Okay. So if you look at the conditioned medium, there's tubular formation, but if you look at the first flow-through fraction, there's nothing. It's at the level of the negative control. And that you could interpret that to mean that you're removing EVs and you're removing the biological activity, and therefore the EVs are doing something. However, what I found is that the more flow through I corrected in later fractions, the bioactivity actually went back up again to a level similar to that of the original conditioned medium, despite the fact that all of these fractions have no EVs in them whatsoever. And I decided to look at the soluble factors, and so I chose VEGF. I chose VEGF as a representative. It's not the only soluble factor that's likely to be important. I chose it as a representative because it's well known and well characterized, and you know, I had to choose something. And what I found is that in the initial flow through, VEGF is completely depleted, and then it rises again in a pattern similar to that of the bioactivity of the in vitro assay. And this is unexpected because VEGF is a 45 kilodalton homodimer, so it shouldn't be going through that membrane. So there's some kind of off-target artifactual thing happening with the 100 kilodalton membrane. So what I thought could be happening, let me just skip my slide. What I thought could be happening was that there might be some kind of off-target depletion by this method of removing EVs. I suspected that what was happening was basically binding of low abundant soluble factors to the membrane, meaning that they're removed from the flow through even though they should be passing through. And if that's the case, I should be able to passivate the membrane by blocking it with a huge amount of another protein, just like you would with the Western blot, and maybe that will reduce the reduction. And that's what I did. So I blocked them with BSA, and again, you still maintain complete exclusion of EVs. There's no EVs detected in the flow through in any case. However, what you do see is that compared to about an 8% recovery of VEGF in the flow through from non-passivated filters, when you passivate the filter, you recover almost 60% of it. So that there was some kind of artifactual reduction of soluble factors as a result of this. And when I went on to look at the bioactivity as a result, you can see that if you use passivated filters to prevent this off-target depletion, you get a complete recovery of the pro-angiogenic effect, despite the fact that EVs are completely absent. So this would indicate that in our experimental system with our MSCs, cultured in the way that we culture them and used in this in vitro assay, EVs are not necessary. The absence of EVs does not affect the strength of signaling. So we then come on to the question of necessary versus sufficient. So I would argue that the data I presented here shows that in our system, and I emphasize in our system because there's so many different tests for MSC-EVs and there's so many different cell sources, it would indicate that they're not necessary. But that doesn't mean that they're not sufficient. So you could have two completely independent arms of signaling, one by EVs, one by soluble factors, and either one of them could act independently to suffice, right? So it's possible so far that if I were to isolate EVs, maybe they can still be involved significantly in signaling. And so what I wanted to ask was, what was the relative contribution of EVs and soluble factors? So typically, it's quite difficult to assess this at the same time. Generally, you will look at the EV output of your cells and you won't necessarily be aware of the soluble factors they're manufacturing because you discard them during the purification process. And that's especially the case if you culture in anything other than basal medium, because of course you've got whatever it is in your medium, kind of muddying the water. So what I did was cultured my MSCs in basal medium, and I took everything that they were secreting above 3 kilodaltons, I concentrated it down, and I separated it on a sign exclusion column. This is pretty similar to a standard EV isolation by sign exclusion, except that it's a slightly bigger column, and I'm also interested in the protein-containing fractions. So what I found is that EVs broadly were eluting in peaks in fractions 9 and 10, with a kind of a long tail of elution with EVs detectable up to around fraction 19. But the majority coming out in the early fractions and fractions 9 and 10 as you'd expect, just a little bit still detectable in the later fractions. Conversely, when I looked at the protein content of these fractions and the VEGF content specifically, that's all coming off much later, with a peak at around fraction 22, 23. So there's a small zone of overlap in around fraction 18, but broadly speaking, the EVs and the soluble protein are separated by this method. So what I then wanted to do was assess the bioactivity of each fraction, or rather each pair of adjacent fractions, independently. And the reason I wanted to do that was because it would allow essentially a quantitative comparison. Because I think part of the one of the potential pitfalls in this type of research is that we're not always aware of the quantity of EVs we're using and how that relates to the quantity of EVs and the quantity of soluble factors that the cells are making. So what I found is that essentially for the tubule formation assay, there's no significant bioactivity associated with the EVs. It's all associated with the protein in the soluble protein in the later fractions. And something worth pointing out, if I just go back a couple of slides, oops, don't know what I did there. I think it's trying to make automatic captions. All right, I keep going. If I go back, you can see that the protein concentration in the peak slides is around 2 × 10⁻⁻⁻, which is considerably higher than the particle concentration in the original conditioned medium. So the number of EVs in these peak fractions is higher than the number of EVs in the conditioned medium, where the bioactivity was originally seen. I wanted to show that this wasn't just something specific to the tubule formation assay, so I also added a simple cell migration assay, and it's the same pattern. There's nothing associated with the EV peak fraction. It's all in the later fractions where the soluble protein is. This wasn't donor specific either. I did 2 additional donors, so this is the second donor. You can see that the pattern of data is pretty much exactly the same. If anything, it's slightly nicer. And it's the same story again with donor 3. And they're all from different ages, different sexes, different ethnicities. So the conclusions I drew from this, and again, this is in my experimental system with my MSCs, my medium, my testing, that EVs in my system were neither necessary nor sufficient. They basically don't seem to be doing anything. The average concentration in the peak fractions was higher than the concentration in the conditioned medium to start with. So given that, is there some situation in which I could have this system in which I'm pretty confident that EVs are not contributing and get a false positive result, which makes it look like they're important? That was what I wanted to go on to. And again, I want to emphasize here, this doesn't mean that, and I'm definitely not arguing that, there are no processes in which EVs or MSC-EVs are genuinely contributing to signaling. It's more that this is what's happening in my system, and I think it possibly has applicability elsewhere. So I think the first and most obvious thing to look at was purity. And I think many of us are aware, I think that there's a lot of discussion to be had around the methods we use to purify EVs and which ones are suitable under what circumstances. But I think it's fair to say that there's a general consensus now that polymer precipitation is unreliable, especially if you don't then combine it with some kind of subsequent cleanup step. It's been shown quite a few times to co-isolate contaminants, both soluble protein and also RNA and other stuff, and of course the precipitation reagent, whatever it is also along for the ride too. So I thought, let's try it with a purification method that's likely to co-isolate contaminants and see what happens. And what I found is that when I took the same starting material that I used for the previous experiment, and I basically concentrated it down and then followed the instructions for the, the commercial precipitation reagent. I'm sorry, I think someone's got their microphone on. Okay, I've muted it. What I found is that, as you'd expect, there's EVs in the sample, so you can see the CD63, you can see there's particles, they're of the right diameter, you can see EV-like structures on cryo-EM. But what I also found was that there's a lot of co-isolated material. So although it recovers a good percentage of the particles in the conditioned medium, it's also recovering quite a lot of VEGF. And a big part of this is the drastic decrease in volume that occurs when you do a precipitation thing. So generally, from what I did and what I've seen in the literature as well, is you start off with a large volume on 100 mil, you concentrate it down with an amicon or something similar, and then you do the precipitation reaction, and then you resuspend it again in a small volume. And even though actually about 98% of the VEGF was removed, still, because of this drastic decrease in volume, the final concentration of VEGF was actually about an order of magnitude higher in the final EV sample than it was in the conditioned medium. And that's also the case for the soluble protein. So the amount of protein in this final sample was just under 600 micrograms per mil, which, if you use the Weber and Clayton metric of particles per microgram of protein, comes out to a value that's well short of what you'd expect for good purity. And the reason I'm confident that this is not EV associated is that were this protein and this VEGF specifically EV associated, it would have been detectable in the size exclusion experiment I just showed you. It would have been well within the detection range for the VEGF ELISA we did there and for the protein quantification we did there too. So we have protein and specifically VEGF along for the ride in these samples. They're not genuinely EV associated, they're contaminants. What does it look like? It looks like a great result. So if you have this sample, you get a strong stimulation of angiogenesis at a reasonable particle concentration, and you get the same thing again with cell migration. In other words, if you were to purchase this kit, follow the instructions, and then check to see whether you have EVs and bioactivity, you would get a result that looked great. It would look like a success, but it's not EVs, it's soluble factors. So what else? The second thing I want to come on to talk about is quantity. And this is a slightly harder idea to get across, I think. I think the idea of impure samples with active contaminants is quite straightforward. This one is a bit more conceptual. Generally, when we quantify EVs, we might use protein concentration or particle concentration. It's the case, I think, that it's quite difficult to relate the amount of EVs in your final sample back to the original biofluid or cell culture medium that you originally had. And I think it matters because essentially there's no real standards for how much EVs is too much to use. There's not really a standard for potency yet. And I would argue, if you're starting off with an effect that you see in conditioned medium, and in order to recapitulate that with EVs, you have to use 10 or 100 times more, I would argue that's not a great indication that the EVs are responsible. It should be, in my view, a concentration similar to that at which they were originally present. So, if you don't check to see what the dosage of EVs you're using is in relation to the amount of EVs in your conditioned medium, you're not going to know whether or not you're using a vast excess of EVs or an amount that makes sense. So what happens if you use a large quantity of EVs that are relatively well purified, but perhaps don't do very much? So I basically repeated the size exclusion experiment again that I showed you earlier, but instead I used basically a lot more medium. I started off with about, I think, about 1.5 liters, and I ended up with much higher concentrations, although I had quite a lot of sample loss. And what I found is that it's a weak signal, but at very high EV concentrations, you do get some weak stimulation of angiogenesis. And if you compare this to conditioned medium, in which the particle concentration has been matched by concentrating it and then diluting it again, you can see that for matched particle concentrations, the conditioned medium is far more effective at stimulation. I would have liked to achieve even higher concentrations to test those, but I kind of ran up against the limits of what I could actually grow and purify. So that's it for my actual data. I hope it's okay. The next few slides are going to be a bit wordy. I just want to go through what I think is going on and what I think perhaps can be done about it. So to summarize our data, in our experimental system, EVs weren't necessary or sufficient. They basically didn't appear to do anything. However, you can get false positives from off-target depletion of non-EV factors. So the ultrafiltration experiment that made it look like EVs were essential, but actually it was off-target depletion. A ineffective purification method can give you great results, but they're not real. And I would argue perhaps an even more difficult thing to deal with is if you use EVs at concentrations that are too high to make sense, I would argue that you're not necessarily recapitulating the actual effect you originally observed, you're chasing something else. And it could be the presence of small amounts of active contaminants in the EVs, or it could be that the EVs themselves are genuinely able to initiate signaling, but they're not actually as effective as everything else that your cells are pumping out. So I think the questions are, how widely does this apply to existingly published HMSC-EV studies? And for me, I would say that the fact that there's not really a consensus on the mechanism between different labs indicates there's some kind of uncontrolled confounding factor going on. That, to me, indicates potentially that there's some kind of issue with co-purification of other factors. I also think that this has implications for any studies of bioactivity from EVs from any cell source. And I think in general, the potential of soluble factors to co-purify and then stimulate something should really be a top acknowledgement for risk when we design experiments. So how can this happen? Here's where I see the biggest risk. I see a lot of studies that follow this formula. So generally speaking, a study will start off with conditioned medium or a biofluid, and they will use one of the standard isolation methods, perhaps a relatively good one, perhaps not such a relatively good one, and then they'll get an EV sample, and then all the characterization is carried out on the output of that isolation process. And most of this characterization is essentially positive verification of the presence of EVs. You're looking for EV markers, you're looking for particles, you're doing EM to visualize the EVs, but the only standard negative verification is checking for something like calnexin or GM140. And all that's really doing is telling you whether you've got contaminating membrane from other cell membrane populations. It's looking to see whether you've got bits of Golgi or bits of ER. I would argue that by far the greater risk is the presence of non-EV material that is soluble contaminating protein, that is active. These studies don't check for that, and I would argue that they're vulnerable to the kind of false positives I've described today. So how can this happen? What can go wrong on the field of purity? It's partly the fact that there's this initial experiment to remove EVs and check that removing EVs matters is almost never performed. I see it very rarely. And as I said before, most of the characterization is just to check that the EVs are there and that they seem normal. The negative verification is relatively, is not done quite as much. And I think in general, there's also a more subtle point, which is that some isolation processes obviously have a much higher potential to be effective than others, but there's also sometimes an assumption that they're working effectively. So for example, density gradient can work great, but that effective function is a ceiling, not a floor, excuse me. And unless you check, you don't know, that your pellet was resuspended without soluble factors coming in, or that your gradient is intact, or that you haven't got something else there that you shouldn't have, even though you don't expect it to be there. And on the other hand, with quantity, as I said, it's really about this concept of potency. Where does the effect that you're looking at, is it relevant or not? Is it the main thing that the cells are releasing or not? And I think because there's not really any widely accepted standard for potency, it means that optimization can sometimes be performed with respect to function. In other words, you might adjust the isolation process or the quantity of EVs you use until you have a working in vitro assay. The problem with that is that you could just be adjusting your processes until you've got a decent concentration of contaminants that are actually doing something. That's my concern here. And the other thing is, as we all know, it's quite difficult to establish that EVs are doing something, because unlike, say, establishing a protein, you can't knock out an EV. You can't remove it and see what happens. You can, there are some inhibitors, but they're so, I would argue they're so toxic that it's quite difficult to be certain that it's due to the on-target effects. And as we all know, it's hard to get a high quantity of pure EVs. There's often a trade-off. So I'm coming up to the end. What I suggest we do about it, I think really the most important thing is to consider that this could be happening. I think that's really the main thing. And then what to do with it, I have some suggestions, but I think that also might come well from discussion within the field. I think there should be wider use of experiments where you remove EVs to see what happens, and you check that removing EVs doesn't also remove something else. I think there should be a much more concerted effort to make sure that there's not non-EV material in your EV sample. I think I'd like to see more wide use of the particle to protein ratio that Weber and Clayton reported in 2013. I really would like to see an end to pollen precipitation as an isolation method. I think at this point, it's really just sketchy. If you use something else after it, some kind of cleanup method, that's fine. But as a standalone method, I think the evidence has really mounted up. And finally, I'd like to see some kind of discussion around EV doses and EV potency. So I think I'm coming up to the end. Just want to thank my co-authors, Annika, Valeria, Ulrika, my PI Molly, my funders, the EPSRC and Rose Trees, and thank you all very much for your attention.
And thank you, Tom. I want to say thanks a lot for this interesting work, this very provocative work, also provocative conclusions that you've shared with us. I think you've given many people a lot to think about. So,
well, if I could, I'm really, and this is something that came up in the reviews, I'm really not trying to be provocative. And I want to emphasize that I do think that there's still very much the possibility for MSC-EVs to be functional and important in signaling. It's more that I had a situation in which I found that they didn't, and I wanted to look at the kind of experimental artifacts that could trip you up. And I kind of see this as a progression of a lot of the work that's going on to look at how isolation processes can impact results. I think it's really a progression of that, and that's my view.
Right. Yeah. So I agree with you, and I think that's precisely why I call it provocative, because I think that we all need to think about these issues, whether we're working with MSC-EVs or with other EVs. So it's important to keep these possible artifacts in mind. And I see that actually I'm a bit overwhelmed here because we've had a lot of questions come in the chat box. And I'm not sure how best to try to address these. So we're even though we have half an hour, we may be hard pressed to get through all of the questions. So I would like to propose that if we don't get through everything, I will have a record of those questions that are asked, and I can share those with you, Tom.
Yeah, that's fine. I'll do my best to get to them after the presentation.
But let's see what we can get through right now. And I think, again, in light of the sheer volume that we have here, I'm just going to go through start at least to go through in order. And what I will try to do is to unmute the people who have asked the question, so that we don't also get overwhelmed with multiple people trying to talk. So we'll start with Patrick James. So Pat, I know you had a few questions that you had here. So why don't you go ahead and ask those of Tom.
Okay, thanks, Ken, and thanks for the presentation, Tom. So I guess the first question I had was just confirmation. It sort of comes down to something that you sort of mentioned on one of the later slides. You confirmed that you actually had EVs in all the fractions. So did you do a Triton treatment of the fractions?
So there's EVs, there's detectable EV markers in fractions 9. Most of the EVs are in fractions 9 and 10, and there's some detectable marker up to around fraction 18, 19. I didn't do any Triton. The markers I was looking at were external membrane markers.
Yeah, so I guess the question comes down to, are you sure that they are actually on the EV, or whether you're having protein aggregation? So that sort of came to the second part. How do you treat conditioned media? So if it's frozen, the conditioned media that before you run it down SCC. So the question will tell you that, say, CD63 is there, but is it still associated with the EV, or is it precipitated, aggregated?
Well, the CD63 elutes in the early fractions, and it elutes alongside NTA verified EV, well, particles from NTA, particles of the expected size, and the expression of those EV markers, the strength of that signal correlates with the particle number.
I guess if I just ask the last question that I put up, Ken. So you mentioned that PEG, and I agree, PEG isn't a great way to purify EVs from contamination. But I guess the challenge that the field faces is, once you do the research, you want to translate it into a therapeutic. So if we want to put PEG on the shelf, what sort of scalable purification method would you suggest?
Well, I think you bring up a great point. And the point is, on the one hand, we can talk about purity all we like. On the other hand, if it works, what's the problem? And I think, you know, if you, so you think that PEG is perhaps the better option for EV purification for therapeutics?
No, I think PEG's terrible. So I agree with you on that point. I think PEG's terrible.
Yeah. I mean, I'm not an expert in this kind of scale-up debate. I understand, I think that people are favoring TFF for this kind of thing. And I think that may have the potential to give a decently pure output. I think basically, obviously there's an issue of scale. I think for me, the important thing is if you're going to bring a product to market and you're going to say the active ingredient is EVs, you can't be putting in something that's EVs and contaminants. And I think if you want to bring in something that's perhaps not the highest possible standard of purity, perhaps you should open the discussion to be about, okay, it's multiple things in the secretome. We don't quite know which is which that's important.
Yeah, indeed, that's a great point. You know, I think that there's nothing wrong with PEG at all if what you're trying to do is get everything that's present in the secretome. So in that sense, you know, what we really want to be sure of as biologists is that if we're talking about mechanisms of action, we are actually pinning those on one part or another of the secretome. And so I think in that sense, you know, if you think about what I guess Bernd Giebel and Sai-Kiang Lim and some others, you know, from some of the isoposition papers have suggested that we call these things EV preparations rather than EVs, and recognize that there are other factors in there. Because in the end, if we want to treat a patient, we want to make sure that the product is it works, and whether it's the EV or not. But you know, if we do need to say what the mechanism of action is, then that's important. So, did you have a follow up on that, Pat?
No, I think there's so many other questions, Ken. I think I should yield the microphone to everybody else.
All right. So I have a question that came in from Yvonne, I think, who does not have her microphone working. So let me just I'm going to go back to full screen here so we can see everybody. So Yvonne asks, all right, you might use EVs at higher concentrations than necessary, but most MSc papers seem to be about therapy. So surely bulk delivery is good. And for physiology, cell culture is a challenge anyway, because in vivo you get constant delivery over a period of time. So how do you get around that difference when you're, you know, comparing the in vivo, I guess, cell administration with what you're collecting in culture?
This is a great point. And for me, it's about you start off with your conditioned medium from MSCs, and you're saying there is a therapeutic activity in this conditioned medium. And I think kind of the argument to say it doesn't really matter how pure the EVs are as long as it works, I think what may be missing from that is the possibility that it's the soluble factors. And if we're going down the route of it's the EVs and it doesn't quite matter how pure they are, well, actually perhaps, let's say hypothetically that the EVs are less important, why aren't we focusing on the soluble factors instead? You know, if we are suspecting, because impure things work better, that the soluble factors are contributing, why aren't we just looking at the soluble factors? That's my take. When we're looking at EVs as a therapeutic, I completely understand the argument that what matters is whether or not it works. My point is that if we suspect it might be working not just because of the EVs, we should take a direct look at that, and we should consider the possibility that the soluble factors that may or may not be coming along with the EVs are actually doing something pretty significant.
Great. Very good. All right. All right. So I'd like to go back to, I think we have quite a few technical questions too. So, John Sperduto has several of those. And so John, I'm going to try to unmute you here. So go ahead. Did it work? Unmute. John, I can't seem to unmute you.
There we go.
All right. We can hear you.
All good? Okay, great. I'll keep most of my questions offline because they are pretty technical and I want to give other people a chance to discuss. Really, the only thing I'd like to raise is coming to the EV world as a mechanical engineer primarily during my undergraduate. I always looked at ultracentrifugation with a bit of an eye of skepticism, knowing how that would work in terms of transport. It's very easy as a dead-end method with high forces to co-precipitate proteins. I would love to see what you did here applied to ultracentrifuging, because I think that's a major, not hand wavy, but it's still a very open question. How can not just lipids but also proteins co-precipitate under such high forces when you have very concentrated culture media and other biofluids?
I completely agree, and actually I would have loved to add that. And the main reason I didn't was a question of time and technical issues. We had an issue with our ultracentrifuge where it had some kind of weird vibration and nothing would pellet properly. And for the sake of the study, we just ended up not doing it. But if I were to do it again with a functioning centrifuge, I would have loved to do that too.
Awesome, thank you. Yeah, I'll send you an email afterwards and we can talk more if you have time, obviously.
Yeah, no, I'm happy to talk. I just might take a while.
Awesome, great study. Thank you.
Good, great. Thanks for the question, John. We have a few questions that I think I'm just gonna throw in here myself. So the MSC source, where were the MSCs from?
These were commercial MSCs from bone marrow that we got from Lonza. They were, I think there's a there's a table in our methods of the donors with their sex, ethnicity, and age, and a reference number. So you can look them up online if you want to.
Okay, so that does answer some of the other questions that have come in too about the source of the cells. In passage number, what about passage number?
We were using them. The expansion was between passage 4 and 6. So by the time we were isolating the EVs, they're at P6.
P6. Okay, good. So, and from what I understand, you did the tubule formation. You did not do other assays, and that's why you're saying that you want to make clear that this is in this particular system that you were using.
Well, no, I did also do the wound healing assay, and the reason I added that was to demonstrate that it wasn't just some quirk of the tubule formation assay. I've got a cell migration assay in there as well, and that was in the slides too. I wanted it to. I added that basically to show that it was an issue surrounding the purification and quantity of the EVs, rather than some weird little quirk about Huvex on Geltrex, basically. So the migration assay is a different cell type, a different behavior.
Mm-hmm. Okay, good. All right. So I'm going to go to Craig now. Let me see if Craig is. I think Craig may have left us, but had an interesting question. Or no, just a minute. There we are. Craig Ayer, you had I think a very interesting question here.
Hi, I thought it was a great talk. Thank you. While you were sort of wrapping up your takeaways from this, one of the things that you were talking about was the concentration of the EVs that are in the original biofluid and not having a standardized dose for what we would expect to deliver to see therapeutic benefits or to observe the phenotypes that we want to observe. My question is mostly about the concept of dose and standardizing that. In an in vivo system, we would have a cell producing EVs over some duration of time, either following a stimulus or constitutively. So when we're talking about purifying EVs and applying them in doses that are much, much higher than what they were found in their original biofluid, is it possible that we're recapitulating, say, a high dose of EVs that would ordinarily be given over a long duration of time that we could see in an in vivo system? It's sort of like, what would you think about the relationship between an instantaneous dose at a high concentration versus a long duration dose at a lower concentration?
I think that's, in a way, it's quite a difficult question to answer, and you're right to bring it up. The concept of dose that I was referring to, the reason that there's no standard yet is because it's a hard thing to try and get right. And this was really only applicable in a situation where you're starting off with the conditioned medium. So you've grown your cells ex vivo, you've not put the cells in. You've grown your cells ex vivo, and then you're looking to see what the conditioned medium does. And I would argue, if you put your conditioned medium into the heart with X number of particles in it, and it does something, and then you purify X number of particles and put it into the heart, and it doesn't do something, under those circumstances, you should question whether the EVs are functional. I think the question of dose over time versus a bolus dose is interesting, but in a way it's not I'm not sure what the answer is, and I'm not quite sure how to conceptualize it. Again, I think there's the question of are we seeking a therapeutic, or are we trying to find out what's genuinely responsible out of all the things that the cells are making for what's going on. Yeah, you could argue that if you're looking at EVs secreted from cells versus EVs injected all at once, that there is an argument for putting a lot of EVs in at once, but I would argue also if you're putting in cells, they're not just releasing EVs by any means. So it's, in a way, I wouldn't know how to do that comparison in a way that was meaningful.
Right. Yeah. Teasing out the effect that way would be incredibly challenging because it's not just dose, it's not just bioactive, it's how long, when, where. Yeah.
Yeah. Yeah. And it's just so many uncontrolled variables. I would say that the concept of dose kind of has to, the way I've thought about it, the only way I can see to think about it is that it's from EVs that are made in vitro.
Sure. Thanks very much. Appreciate it.
Thank you.
Thanks for the question. I want to go next to Benedetta Busalati. Benedetta, you had a question, and specifically about some other materials that can be involved here.
Oh yeah, thank you, Tom. Very nice presentation indeed. So my question was, did you have the chance also to take into account the role of microRNAs that we all know are contaminants?
So no, sorry, can finish your question.
Yeah, I understand that you might have some ideas and your opinion on that. And then I also have a more general comment in the sense that we all know that EVs are very sticky, especially considering DNA and microRNA fragments. So how should we consider that? As a contaminant or part of the EV cargo?
Interesting. Well, for your first question, I didn't really look at microRNAs. I didn't look at microRNAs at all in this study. It was more that I was chasing the function, and I was finding out where the function was and whether that was associated with EVs or soluble factors. And because I found that the EVs weren't doing anything, I didn't bother to look for microRNAs inside them. Definitely, there's been a lot of good work by other labs to demonstrate that you get co-purification of microRNAs with various methods. Again, the polymer precipitation methods are pretty bad offenders for that. I think that's both going to confound, well, I think actually that could be quite a large reason of why it's so common to identify EV-associated microRNAs as the causative factor for EV signaling. But yeah, microRNAs as contaminants is certainly something to be concerned about. And your second point, would you remind me? Sorry, what was your second question?
Yeah, it was just more than a comment saying
EVs are sticky. Yes, thank you.
What outside EVs? Yeah.
Well, so the question is, what kind of interaction between an EV and a soluble factor is meaningful, and what kind of interaction between an EV and a soluble factor isn't? And that's difficult to answer because we're asking questions about what may or may not happen in vivo, and I would argue we can't actually really know. I don't know how I would show whether an EV was associated with delivery of some kind of other soluble factor externally in vivo or not. I would say that if you can remove it from an EV by pretty gentle purification, it's not meaningfully associated. So in my example, if after sec, which is basically just the EVs are in PBS for a bit, if you think about it from their perspective, and then there's no more protein or VEGF associated with it, I would say the protein and the VEGF isn't associated with it in a meaningful sense. I think the context of that stickiness has to be, would it be reasonably expected to occur in vivo? And moreover, is that stickiness just something, are the EVs helping deliver the factor? Like, does the EV make the uptick of that factor more effective, or does it just mean that that factor is in your EV tube after your isolation? So, you know, if you've got something on the outside of an EV, and because it's on the outside of an EV, it goes into a cell better, then you can argue that it's important. I would say if it's on the outside of an EV, but it doesn't really make a difference or it hinders uptake, I would argue that perhaps it's not important, especially if it's removable by purification.
Yeah, okay, the point of purification, I think, is fair enough. Yeah, okay, thanks.
Thank you.
Thank you, thank you. Chiro Teta, I would like to unmute you now because you make a good point about the regulatory agencies and how we need to speak with them. Chiro, are you there? Having a problem unmuting people for some reason. Sorry. You know what? I'm just gonna let everybody unmute themselves at this point. Chiro, are you there?
Yeah, I'm here. You hear me?
Yes, I hear you.
Now the point, well, first of all, thanks so much. Found it extremely interesting. But I think the interest from regulators is particularly when you come up with a final documentation for EVs as the active product. So the question, I mean, they will certainly read your papers. And once they read your papers, they will have a kind of confusion. Are we talking about EVs, or are we talking about EVs in addition to something else? And if this is the case, the opinion from the regulators would be that in coincidence to a submission for EVs to be applied in clinical medicine, they would argue that it's the EVs plus something else, or even worse, it's not the EVs at all. So I just want to put in a positive way, but I think these studies really emphasize the need to further develop all strategies in order to not to be let down by the regulators. And you know, the regulators are people who need to know if you are making the right product for the right application.
I agree. And I think actually the regulators would be correct to take that stance. I think that if we're going to bring a product to market, if we're going to claim that an EV is therapeutic, we really should be pretty certain that it's the EV. If we want to make a different claim and say that we've got some kind of secretome preparation and that's beneficial, we don't know quite what it is, I believe there probably is a route for that, because it's the same with cell therapeutics. But I would think that it is going to be an issue trying to bring an EV prep to market if we're not very confident, or if there's some uncertainty that the EVs themselves are not the active factor, regardless of whether it's functional or not. Yeah, I don't know what to say other than that I agree, and I think that might well be an issue.
Thank you.
I think the way forward would be, you know, to perhaps do some of the stuff I talked about, be much more active in seeking contaminants, be much more active in doing experiments where you remove EVs to make sure that it matters. And if it turns out that it's the EVs and the soluble factors, maybe we need to pivot and acknowledge that they're important. Maybe that's the way to go for therapeutics.
Yep, I agree. Thank you.
Good points. Thanks for making those. So we have Leonid Margolis is with us today, and so he's done some studies on cytokines associated with EVs. So Leonid, I'd like to just invite you to bring up your question to Tom, please.
Thank you, Ken. I'm echoing the previous questions and points which Ken made in his comments. So there are two different issues. One is the mechanism of the phenomena, and one is whether we use it for therapeutics. For example, you say that it might be cytokines, as I know in some other systems, which do physiological effect. And some of the cytokines are attached to extracellular vesicles. So that's probably when you have high unphysiological, what you call high concentrations of EVs. They also work the same way as soluble fraction. So if you want to do mechanisms, they may be both important, and EVs carry much smaller amounts. So you need this unphysiological, I would say, concentration. But for therapeutics, of course, maybe EVs are being more profitable, like they can be preserved for longer times. And that may be finally the preparation which will be used therapeutically. Also, we don't know locally in the tissue, which you started your presentation with, how much, what's the concentration of these EVs close to the target cells. So both of these can be true, and the mechanism is one thing, and you made your points, Tom, very well. And the development of some products for therapeutic is another issue. It might be still these.
No, that's valid, but I think if we're talking about developing products for therapeutics, say in this example, so in this example when we're talking about cytokines associated with the outside of an EV, if the activity is due to enhanced delivery of the cytokine because it's on an EV, we should be clear that that's what it is, that it's the cytokine's action and it's enhanced delivery by the EV. And in general, when we talk about the EVs are active, I think we need to be more precise. We need to acknowledge that these things are potentially in our final samples, and they may or may not be. It may or may not be the EVs themselves. In my system, for example, I didn't see any attachment of anything to the EVs. There was no protein that I could detect. There wasn't any VEGF. It may well be different for different systems, but the concern I had was that if you used a poor purification technique with my system, you would conclude that there are things like VEGF associated with the EVs, but they weren't actually associated. They were just in the same tube. And that's what worries me, and that's what I wanted to talk about. But yeah, as between the question of mechanism versus therapeutic, I accept, of course, the point that fundamentally we just want to find something that works. But I don't think that it's quite necessarily that easy to disentangle. I think, as it's been mentioned before, if we're trying to bring something to market, if we want to get it into patients, we need to be precise about the mechanism, and we can't not acknowledge the presence of non-EV factors in the sample, especially if they appear to be important.
Yeah, thank you. You made your point very clear, except the EVs can carry, as was mentioned, RNA or something else. It's still EVs. It depends on how we define EVs with everything which they carry inside, on the surface, bound.
Yeah, no, you can certainly, I have no problem with saying that stuff associated with the outside of EVs is part of an EV signaling complex. I think that's completely reasonable, and I don't disagree. My concern is that with the wrong methods, you might wrongly identify something as part of an EV signaling complex when it isn't.
Yeah, thank you. It's very clear you made your point. Thank you.
Thank you, Leonid, too. So we're coming towards the end of the hour, and I'm very sorry that we don't have more time. But I wanted to give a word to Bernd Giebel as well. Bernd, would you mind just unmuting yourself? And I know you've had a lot of comments here, but maybe in the chat box. But do you have some last words about things that we should be thinking about with soluble versus EV associated factors?
Yeah, thank you, Ken. And Tom, it was a great talk. We chatted already via Twitter. I think, yes, what we have is not an isolated EV product. So we always have components co-purified. So we use PEG and wash it away with ultracentrifugation, but for sure there are other components. LDL is their lipoprotein particles. So if you do ultracentrifugation, you will have other byproducts, and I think nobody ever has a pure EV preparation. And thus we never know, is it really the EV, which is the active component, or is it the byproduct, or is it modulated by the byproduct? To learn whether EVs are the active components, we need to destroy them without removing them, because if you remove them or wash the samples, then you may remove other active components. I don't know how to destroy EVs, but with detergents, and then they kill the reporter system. So that is one thing. And I think what we can do is we started to discuss about cellular cell therapies with MSCs. And you can now apply MSCs as self-replicating cells, maybe forming tumors. And the field now manages to concentrate something, let's say something from the supernatant in a very reproducible manner. And we can characterize it, and we can have molecular fingerprints. And with the molecular fingerprints, we can see whether we can produce it reproducibly. And if that is what we get is helpful without showing any side effects, without knowing the exact mode of action, I think that's an advantage for the field. Yeah, so like aspirin. So I think it has a lot of clinical potential. And are we aware about the mode of action? So we need to be careful from the academic side whether we are too critical. So I'm a basic researcher and I try to understand how MSC-EV works, whether it's indeed MSC-EVs, but on the other hand, we have started to translate and it's two different words. Yeah.
Well,
yeah.
I do agree with you, and this comes back to the point I think a lot of people have touched on, which is what really matters is whether it's a therapy and whether it's reproducible. And I completely take your point. We'll never really have a truly pure EV population, and it is very difficult to remove or destroy EVs without in some way affecting the rest of the sample. The only thing I would say is that imagine a situation perhaps in which it was the soluble factors that were mostly important. How would we distinguish that from a reproducible solution that contains EVs, but the EVs themselves, the actual liposome-like particles, are relatively not contributing that much? How would we know whether or not that was the case? Is it actually that this concentrated supernatant we have after the end of it is mostly something else that's functional? How do we distinguish that situation from a situation where the EVs are really contributing? And I want to emphasize again, I'm in no way saying that I think that EVs don't do anything. It's more that I had this experimental system in which I found in my specific setup that they weren't, and I wanted to see, given that, is there a way we can be tripped up? And the purpose of this study was to identify things that we might be being tripped up by. It's not to argue that MSC-EVs never do anything, because I couldn't possibly argue that based on the data I have.
I like to give you another thing. So it depends on the assay. How fair is the assay? What is the assay reporting? Is the tube formation assay the right assay? So we think on immune modulatory assays. Are they the right assays? I don't know. What I can tell you, when we treated the GVHD patient already with a 30% considered dose, we saw an effect. And to see effects in any in vitro assay, we have to concentrate and apply the sample which we apply to the human 10,000 times higher. That means we're in vivo. There is a completely different thing, and it's much more sensitive what we learn. And again, we think we have good and fair immune modulatory assays. I don't know whether it's really these which makes the effect. With that, I agree. But in vivo, it was 10,000 times more sensitive. And yeah, that is what is the system? What is the appropriate assay? And that is a very important thing. In your essay, I agree, it was convincingly that it's maybe not the EVs, but maybe you have other assays. And again, final proof you should get from in vivo data. So you need appropriate preclinical models which you apply the different samples. And we agree and we discuss MSC-EV preparations act multimodally. So there is not one mode of action or one mechanism of action, that they are different, and it's an integrated activity. And I really consider that indeed what you nicely showed, soluble factors which are co-purified may really essentially contribute to the therapeutic activity.
Well, it's very interesting that you have a much lower requirement for sample in vivo, and definitely I would have liked to have in vivo in this study. I think the focus for me was more about not trying to prove that EVs do nothing, but trying to demonstrate that in a system where the apparent activity of EVs was not real, could it appear real? So I can't make any argument based on what might have happened had I taken these samples and put them in a rat, because I just don't know. And you raised an entirely valid point about in vitro versus in vivo. This is why I'm not trying to argue that MSC-EVs are not functional. It's more that I'm trying to say, in this in vitro experiment, when you control for these things in a way that hasn't been controlled for before, EVs don't appear to really matter. However, if you if you carry out your experiment in this particular way, in a way that's quite common and entirely understandable, you can get false positive results that implicate EVs in these same in vitro experiments, whereas we know actually the EVs aren't important in these in vitro experiments. So the focus of my paper is more on the technical side of things, because I agree that to make conclusions on the in vivo activity based on this data, or the kind of general therapeutic activity of the EVs would be inappropriate. It's more that I've identified things that I think could be a risk, and whether or not they apply in vivo or whether or not they apply to preclinical studies, that's going to be very much dependent on what people are doing in their own labs. It's more that I want to bring these risks up so people can think about them.
No, that's absolutely clear, and I think that is what we need in the field, to be more critical from our own side. And there are other trap mines, and we can discuss for hours. And I really appreciate that study, and I think that should sensitize us all. And yes, we should discuss it. And yeah, we discussed it in some positions papers before. Ken is also, I think, on the latest, you were the first author where we discussed the identity, and we discussed the issue whether there are byproducts, and this is exactly what you show. You confirmed our things. You should have published 1 year before, then we could have cited you.
Well, certainly I think that we've had a lot of good discussion here today, and we've also learned how much more there is to find. So thank you so much, Tom, for sharing your work with us. I think that we do have, Alex Mitzialis is also saying here in the comments box that the regulators are going to be most interested in what happens in vivo, and there is, good evidence that EVs are acting there. So it's certainly not the case that they don't act at all. And Tom has made that point too. So we just, we all, I hope, as we take things away from this journal club today, we'll redevote ourselves to rigorous and reproducible science. As we as a community are certainly interested in already. So thank you all for joining today. Thank you, Tom. I'm going to collect all these additional comments, and we'll see what we can get answered.
Looks like there's about 50 of them. I will try to get to them all, but
we might have to do a stipend or something for this. Yeah. So anyway, thanks again, and I hope everybody has a great rest of the day. And I look forward to talking with you again soon.
Thank you so much for having me. Thanks very much for coming.
Bye now.